Simon. Hope you're having a great DCAT week. Can you introduce yourself for our audience?
Yeah, hi, I'm Simon Keane. I'm VP of Cell Line Development at Abzena. I sit on the scientific leadership team at Abzena to help talk to customers and understand what their needs are and how Abzena can help them.
Could you just provide an overview of the AbZelect platform?
Yeah, the AbZelect platform is Abzena's cell line development platform for the generation of high-producing clones to make therapeutic proteins. It's based on the CHO-K1 system, and we've developed really so that we can quickly and reliably identify clones that are the right clones for that product. And the right clones are those that are high-producing, that generate good quality product, will ease the development pathway and ease the regulatory pathway. And it's a cell line that you want to live with your asset throughout its life cycle.
Can you give us a use case of what this enables clients to achieve?
Yeah, it's aimed really at helping to get clients from that important value inflection point of lead candidate selection up to first-in-human trials, IND approval and first-in-human trials.
What are the key improvements in technologies and processes from Abzena's previous CLD approach?
Yeah, cell line development is really based on three legs. It's a stool with three legs, we say. The first is the vector. So we've done a lot of development around improving our vector technology, changing the selection process from DHFR to a GS to give us higher producing stable clones. We've developed the cell line so that we've identified a subclone from CHO-K1 which grows to high densities and with reduced doubling times, so we can increase the productive phase of the culture. And we've also looked at that process. We've changed the media. We've found a media that's more amenable to scale up, so we can more reliably scale up. And it gives us another increment on productivity. So we're seeing those incremental increases in productivity from the vector, from the cell line, and from the media that we're using, and altogether those add up to a significant improvement in titers.
So when you're partnering with a CDMO offering a cell line development platform, what kind of questions are you getting? And building off of that, what kind of questions should you be getting?
Yeah, I think one of the top questions we get asked is about the productivity, about the titers that our cell lines develop. And I guess that's aimed towards understanding what are going to be cost of goods as people go into commercial phase. But maybe it's not quite so important at those early stages of clinical development. I think customers should also be asking the CDMO about what's their track record. What's their track record in general in getting things through regulators and understanding the regulatory pathway. But what's their experience with working with different types of molecule. If you're working with a standard monoclonal antibody, then most companies can deal with that. But if you have a more complex protein, maybe in a novel fusion protein or a protein nanoparticle, then understanding what's the track record around that. And what's the reliability in terms of delivering on timelines and building a package that is deliverable.
When we're at DCAT week, we get this great, all these minds are coming together and there's so much, deals, innovation, what have you going on. Can you give me your bold prediction 10 years from now? Where do you see the future of biopharma?
Yeah, I think biopharma is just getting more complicated. So we're not just using single specificity monoclonal antibodies anymore. We're getting into bispecifics, trispecific, multi-specific antibodies. And we're seeing more of a trend for fusion proteins onto that. So it might be an antibody fused to an enzyme, or it might be an ADC conjugate. So there's actually different types of bioconjugates. So different payloads, oligos, etc. So I think what used to just be a single molecule with a single target is going to be a more complex molecule with multiple modes of action.
Simon, thank you so much for taking the time to speak with us this DCAT week.
Thank you very much. It's been great speaking with you.